science friends. We are going back into genetics, and so everything that we have learned about so far has been in a pattern of inheritance known as complete dominance. And so when we've talked about our genetics and we've done our Punnett squares, we have three different genotypes that you can get. You can get big A, little a, and little a. And in complete dominance, that big A, and big A, little a have the same phenotype. These phenotypes are indistinguishable. That is what complete dominance is. The dominant, if they have the dominant, then they show the dominant trait. If they have two recessives, then they would show the recessive trait. But you can't tell these two apart. And that is a complete dominance set of inheritance. Everything that we have done to this point has fallen under that pattern, okay? But there are many traits that aren't completely dominant over the other. Actually, most traits do not follow that pattern of inheritance. Sure, there are several that do: widow's peak, hitchhiker's thumb, attached earlobes, freckles, things like that. PTC tasting, all of those things. Either you are or you aren't, but not all things are like that. So we're going to talk about a couple of those other options today. So the first one what we're going to talk about that's new today is incomplete dominance. And so in incomplete dominance, you have neither allele, okay, or letter, the allele, remember, is like a letter, is completely dominant. And so you have a hybrid that has a phenotype somewhere between both parents. So in incomplete dominance, you really have an option for three phenotypes, okay? You don't, versus in complete dominance, you have 1, 2 phenotypes, okay? In incomplete, you have 3. So let me show you an example of incomplete dominance, okay? So in our parent generation, we have the red flower and the white flower, and we mate those together, and in the subsequent generation we have a pink flower, okay? And so that pink flower has the allele, the CW allele from the white parent, and the CR allele for the red parent, okay? Notice we didn't write these like big A, and little a. That would be incorrect for incomplete dominance. In incomplete dominance, we want to write everything as a capital letter because we don't want it to make it look as if something is dominant over the other. Okay, that would be incorrect. And so this is just like they've done here, C, I assume, for color. So it's color white and color red. And those R and W's are both capitalized because neither one is more dominant than the other. And so we have, 1, 2, 3 phenotypes. This offspring, this hybrid in the F1 generation is a mix of both parents, okay? And if you take those offsprings and you mated them, so this one, let's see, if we take the CRCW and CR and CW, so a pink one here, and we mated it with another pink one here, then we can do our Punnett square. We get a red, we get 2 pinks, and we get a white, okay, in the F2 generation. And so you see that incomplete dominant trait, okay? Neither one is completely dominant. Another type that you can have, which is similar, and people have a hard time keeping these two straight, they tend to get them confused, is codominance. And so in codominance, you see both alleles present in distinguishable ways, okay? So if this were a codominant, you would write it the same way. You would write your letters the same way as you do complete dominance. But your flower would look different. Your flower would maybe be white, okay, with red spots. You see flowers like this in the nursery sometimes in the spring. You'll have, white and pink white with maybe pink stripes in them, or, purple on the outside and kind of yellows in the middle. Those would be codominant traits, okay? You see both phenotypes separately. That is codominance, okay? Blood type is another example of codominance, but blood type also, and I'll explain that a little bit more in just a second, but it also exhibits a different inheritance pattern referred to as multiple alleles, okay? So, in all of these examples that we've done so far, we have two sets of alleles. There's one allele, there's another allele, okay? When we did our complete dominance, we had big A and little a. So that was two alleles. But in multiple alleles, you have more than two alleles. And so blood type is an example of that. Blood type, you have I big A, you have I big B, and you have little I. Okay? And so blood type, what blood type does, we always have this red blood cell in all of them that looks the same, okay? O blood type has two little i's. These I big A or I big B code for surface proteins called antigens. I think that says it's, thought I saw that word on here, maybe not. But surface proteins or surface antigens, which give the red blood cell a specific appearance, okay? That's what causes you to only be able to accept certain types of blood, okay? So somebody, like myself, for example, I have type A blood, and so my body recognizes stars. Or triangles, rather. Let's call these triangles. So if you give me a triangle, I know what to do with a triangle, okay? So I can take a blood cell that has a triangle like mine, or I could take a blood cell that was plain, okay? What I can't have is a blood cell that has a circle on it, because I don't know what to do with a circle. My body sees circles and it thinks that is not you, and it's foreign, and it tries to fight that, okay? So these alleles code for these shapes, so to speak. They're carbohydrates or they're surface antigens on the surface of, I think I may have said they're proteins, but they're on the surface, they're carbohydrates, antigens on the surface of a red blood cell, okay? So I big A is going to code for those triangles, I big B is going to code for the circles, and then little I doesn't code for anything. So that's when you have that little I genotype, you don't have any antigens on the surface of your blood cell, and that would give you type O blood, okay? But you see we have three alleles here, and so this represents multiple alleles. It also represents codominance here in this example. In AB blood, you have the circle and the triangle, okay? The IA gives you the circle. The IB gives you, I'm sorry, I said that backwards. The IA gives you the triangle. The IB gives you the circle. And so this shows codominance as well, okay? So I big A, or I big A, little I, gives you A blood type. I big B, or I big B, little I, gives you B blood type. I big B, gives you AB, and little I, gives you O, okay? You can accept what you know. A can accept anything with an A. B can accept anything with a B. AB can accept A or B. Everybody can accept O, and O can only accept itself, okay? Now, somebody may ask, what about the positive and negative? All right. Well, the positive and negative is known as the RH factor, okay? It was founded in rhesus monkeys that are spelled R-H-E-S-U-S. So that's why you get RH factor. And so I'm A positive. So positives can accept negative. Negative cannot accept positives. Okay? Negative cannot accept positive. And that's again another surface carbohydrate or a surface antigen that the cell either recognizes or fails to recognize. Okay? So for me, I'm A positive. So I can accept A positive, A negative, O positive, and O negative. Okay? What about somebody that was B negative? What could they accept? They could accept B negative and O negative. Okay? So today I've got a little blood typing game that you can go online and play. I'm not going to give you an assignment on it per se, so I'll not know that if you did it or not, but it's good practice to make sure, it's fun. I really enjoy playing it, honestly, but it's good practice to make sure that you know which blood type goes with which, because you will be assessed on that later on, okay? That's important, all right? So, AB, positive is referred to as the universal recipient because they can get blood from anybody. They can accept any blood type, positive or negative, from AB, or O. Okay? Which one would be the universal donor? Who can give blood to anybody? What type of blood can anybody take? Because it has the fewest surface antigens. O negative is the universal donor. Okay? If you're a person that's O negative and you ever get in the blood registry, they will hound you to death to come donate blood because that's the blood that they love to stock the blood banks with. Okay? Because when they get low, anybody can take that. Okay? They usually use it in cases of emergency when they don't know and they don't have time to type first. Or they, what else was I going to say? I don't know. They don't have time to type first. Or, anyway, I don't know what else I was going to say. Sorry about that. Okay? So let's look at this blood problem together and see if we can't solve it. So identification bracelets were accidentally removed from 3 newborn babies. Blood typings were taken to help in the newborn identification procedures. The blood types for the babies and their parents were: baby 1 has type A, baby 2 is type O, and baby 3 is type AB. So we have to determine which baby could belong to each type of parent. Okay? So, if Mrs. White and Mr. White are both type O, then they both have to be little I, little I. Okay? So for them, there's only really one baby that they could have. That would be baby 2. Okay? Is going to belong to the Whites because it also has type O blood. And they both only have a little I to give. All right. The next one, let's look at Mr. Black and Mrs. Black. I guess we should have a Mrs. here. Sorry about that. This one could be I big A, or they could be I big A, little I. Okay, we don't know for sure. This one would have to be I big B, or I big B, little I. Okay? Mr. and Mrs. Green, she has to be little I, and he has to be I big A, I big B. All right? So that means their child is going to have to be type A or type B, okay? It would be IA little I or IB little I. So that's going to mean that baby 1 is going to belong to the greens, okay? Leaving baby 3, type AB, to belong to the black family. And so that means that they inherited an IA from mom and an IB from dad, okay? Now, they could have also been, potentially have had O blood type. They could have also potentially had B blood type, or actually, B blood type if they had gotten an IB and a little I here. They could have had A blood type, or they could have had AB blood type. They could have really had any combination with those two parents, okay? Because we don't know their specific genotypes. But that's a sample blood problem that you might have to solve something similar, okay? You actually have a big blood problem today on genealogy of the gods. Some of them you won't be able to narrow down to exactly what it is. Like, for example, you might know that they are B, but you will not know that it's I big B little I or that it's I big B, okay? So you can write either of those. Sometimes for shorthand, you'll also see it written like this, I big B blank, meaning we know it has to have one B allele, and then this one is questionable, okay? So either way you choose to write it, whether you choose to write both or you choose to use the shorthand, is fine with me. Most of them you will know the full genotype. Some of them you won't know the full genotype. You'll just know the phenotype or just know one allele, okay? So let me know if you have any questions on those as you work through them. All right. Let's finish up with a couple more patterns of inheritance. So the next one is pleiotropy. And so that means multiple phenotypic effects, okay? So even with blood type, we're still pretty limited. We basically have 4 phenotypes, right? A, B, O, and AB when we have multiple alleles. But most genes have many phenotypic effects. They can have many combinations, and that is referred to as pleiotropy or pleiotropy, either way you want to say it. And that's what most traits are, okay? Hair color, eye color, skin color. Those are great examples of pleiotropic traits because, you know, your skin is not just, or your hair is not just brown or blonde, right? My brown may be different from your brown, and so on and so forth. Okay, epistasis is another example. Epistasis is when a gene at one locus or spot on the chromosome alters a expression of another gene. So, for example, if you have a trait for albinism, it doesn't matter if your hair, if you're albino, it's not that your hair is, you've got a trait for white on your hair, right? You just have an albinism, and so it trumps that, okay? It's basically the trump card, an epistasis is. It alters the second locus. It doesn't, if you're albino, it doesn't matter if your hair was supposed to be black, red, white, or purple. You're going to be albino, and it's not going to matter because that covers it up, okay? And then polygenic traits. And that also kind of goes to, back to the pleiotropy too, that you have multiple phenotypic effects because you have many genes involved in a trait, and so you have a continuum. You have, not just one phenotype, okay? Because there is not just one gene that controls it. There are many genes that control that, and so it's a combination of all of those things that give you a certain thing. It's like mixing paint at the paint store. Like there's only a few colors that they actually squirt into it, but it's how many drops of each color. And that's kind of how it is with polygenic inheritance. And then lastly is the environmental effect. You've all probably heard about nature and nurture. Even identical twins that have the exact same DNA have sometimes some different traits and or certain predispositions for things. Maybe one of them will develop cancer and the other one won't because of environmental effects. And that's really important as we study genetics as well. Okay, so if you have any questions, then let me know. If you want to do this exit slip for bonus points, send it to me in an email today, Monday, and I'll give you a few points on it. If not, then complete your other assignments, and you all have a great day.